Hey everyone, it's Jamie Batts, your instructor. We're diving into chapter 13, which is an extensive chapter on the brain and the cranial nerves. And if you've looked at this PowerPoint already, you know that it is another very, very long PowerPoint. So I'm probably going to split this video up into a few sections like I've done for some of the other long chapters. Kind of depends on how tired I get as I lecture. Sometimes as I'm just talking to myself here in my office, it gets a little draining just to be talking to myself. So I'll take a little break every once in a while. So if I need a break, I'll take it, but otherwise I might charge on through. I don't know if it's easier for you guys to have an hour and a half long lecture where you just pause it and come back to it, or if it's better to have little sections. It probably doesn't really matter, but either way, we're going to go into the PowerPoint here. This is chapter 13 on brain anatomy, and we'll just dive right in with starting with the basics. So the majority of our neural tissue in our body is centered in our brain. So 97% of all of it packed right in there. Typical brain is about 3 pounds. Size varies from individual to individual, but there's no correlation between brain size and intelligence. Males tend to have a little bit bigger brains than females. Everything in males is a little bit bigger than females. Makes sense. So the brain develops off of something called, oops, forgot to turn my phone off, develops in something called the neural tube. The neural tube is visual by about the 4th week of development, and it's basically make sure they're all turned down, sorry. Basically a infolding of all of the cells that the embryo at that point is made of, and it kind of develops into 3 major regions. These are called the primary vesicles, and they will expand and kind of flower out to form the major areas of the brain. So the primary vesicles are called the prosencephalon, the mesencephalon, and the rhombencephalon. So the prosencephalon or forebrain is at the tip or the anterior end. Mesencephalon is towards the middle, and then rhombencephalon is more called or more towards the tail. That's more continuous with the spinal cord. So here you can see those structures, and this again is just the rudimentary formation of the brain at about 4 weeks of development. So at this point, the mother's just about finding out that she's pregnant. So the brain development by about week 5 is when those vesicles start to change in position. They form what we call secondary brain vesicles. The prosencephalon is going to subdivide into the diencephalon, and that's a major relay center for the brain, and then the telencephalon. The telencephalon eventually is going to form the cerebrum, kind of flowers out and bubbles over the diencephalon. The secondary brain vesicles of the rhombencephalon are going to divide into the medencephalon and the myelencephalon. The medencephalon is going to form the midbrain, the cerebellum, and the pons. And the myelencephalon is going to be eventually develop into the medulla oblongata. The cerebrum will continue to enlarge and kind of develop over most of these structures. So there you see it, and there are some really cool animations if you go into YouTube world, if you're interested in developmental biology, like I am. Basically, the whole structure just, again, it flowers out. It's really, really cool. Just go to YouTube and type in, neural embryonic brain development or something, and there's a ton of really cool animations out there. You can pause the video here, go back and review. Of course, all those animations are above and beyond. You just need to know the information that was on those slides. What I would suggest doing, and I can't believe your book doesn't have a chart like this, but just to basically create a graphic organizer or flow chart of that process of development for each of those ventricles, or vesicles, not ventricles. Let's keep moving. So let's talk about the 6 major areas of an adult brain. So obviously the cerebrum is the brainy part of the brain. This is divided into 2 large cerebral hemispheres, highly folded surface, called the cerebral cortex. And this is our functioning of conscious thought, memory storage and processing, sensory processing, and regulating all of the coordinating activities of our skeletal muscle. The cerebrum has fissures on it, which are very deep grooves. The gyri, which are kind of the mountains, and then the sulci, which are the little shallow depressions. The shallow depressions lie on either side of the gyri. So I remember it by thinking, I jump over the gyri, and I sulk and sulky. Maybe it works, or I'm just crazy. The cerebellum is the little kind of walnut-like structure on the posterior side of the brain. It's partially hidden by the cerebral hemispheres. It's the second largest brain structure, and it's mostly involved in coordinating and moderating our body's position and our coordination. So this is some of our basic movements, hand-eye coordination, and that type of a thing. There you see what I'm talking about. The fissures are much deeper. The sulci are just the shallow depressions, like these little guys. The gyri are the big mounds. And then here you see the cerebellum. Here you can see the diencephalon, the remnants of that early embryonic structure. It's still functioning in us, and it plays a highly important role in relaying information up to the cerebrum. And then, of course, the spinal cord is connected inferiorly. So let's talk about that diencephalon. The diencephalon kind of looks like a little lollipop inside the brain. It has a structural and functional link between the cerebral hemispheres and the rest of the central nervous system. So like I said, it relays information up and down. It's divided into two major areas. The thalamus. The thalamus is our major relay for sensory information, and the hypothalamus, which is mostly involved with emotions, some of our body functions like eating and drinking, as well as hormone production. The brain stem is composed of the midbrain, which has centers for auditory and visual reflexes, and the pons, which has somatic and visceral relay centers in them for autonomic control, like our respiratory centers are in our pons. And then our medulla oblongata, which is, again, relaying information out to the peripheral viscera as well as up into the thalamus and the cerebrum. This is where some of the basic functions of our body, our heart rate, respiration, blood pressure, are located there. Here you see the brainstem and the diencephalon. Again, I told you it kind of looks like a lollipop. You have the thalamus and the hypothalamus, which you can't really see in this picture because you see some nerves coming off of the thalamus here. In sagittal sections, you can see it fairly well. Midbrain, pons, medulla oblongata. So let's talk about the ventricles of the brain. These are formed during neural development in the embryonic form. They're filled with cerebral spinal fluid, and they're lined with special cells called ependymal cells, which you may remember those. They have the cilia to help circulate the CSF. The ventricular system has two lateral ventricles on either side of the cerebral hemisphere. They're connected together as well. There's an interventricular foramen to allow for that communication between the two. Then the third ventricle, which is within the diencephalon, the cerebral aqueduct, allows the CSF from the third ventricle to flow into the fourth ventricle. The fourth ventricle is kind of in front of the cerebellum, like right around the pons area. Again, sagittal section, you'll be able to see what it is in just a minute. And it's a little narrow opening, kind of looks like a sideways triangle almost on most diagrams. There you see it, see that sideways triangle I was just referring to. For that fourth ventricle that leads into the central canal of the spinal cord. Again, CSF continues to flow there. Here you can see the third ventricle. You can see our two lateral ventricles. The interventricular foramen, which is where the CSF flows from the lateral ventricle to the third ventricle. Cerebral aqueduct from the third ventricle to the fourth ventricle. And then on this one, you can see that interventricular foramen even better. So there are connections between the ventricles. The corpus callosum is a tract of white matter that goes in between the two cerebral hemispheres, connecting right and left. And then the septum pellucidum, which separates the two lateral ventricles. So the ventricles are separated by the septum pellucidum. The hemispheres are connected by the corpus callosum. So here you can see what I mean. The septum pellucidum dividing the two chambers, the corpus callosum connecting the two chambers. Notice the distribution of white and gray matter here is reverse of the spinal cord. You have the dark matter. Yes, this is an astrophysiology or astrophysics class. This is our gray matter. The gray matter is on the superior side. The white matter is deep or deep instead of superficial. And that, again, is because the gray matter is all of our cell bodies and the white matter is all of the tracts or the myelinated axon. So that would make sense that that's more towards the interior or deeper layers. You can pause the video here, go back and answer these 3 questions. Oops, went the wrong way. Sorry about that. Let's talk about how the brain is protected. It's protected by the cranial bones, obviously, right? Frontal, parietal, temporal, occipital. There's also the cranial meninges. The cranial meninges are the membranous brain coverings. They're continuous with the spinal meninges, same layers. Dura arachnoid pia, same basic structure. The cerebral spinal fluid is there for extra cushioning. And then we also have the blood-brain barrier, which is biochemically isolating the neural tissue from the circulating blood. So let's talk about those meninges. Again, 3 layers, continuous with spinal meninges. The dura mater has that outer fibrous layer, which is fused to the periosteum of the cranial bones. The inner layer has the meningeal layer. It is more for protection. The layers are separated by a little gap called the dural sinuses. Then you have the arachnoid matter, which has the arachnoid membrane, smooth covering, not following the folds of the brain. Then the arachnoid trabeculae, which is connecting the arachnoid matter, kind of like a spider web, that's where it gets its name, to the pia mater, and then the subarachnoid space, which is where the CSF circulates. The pia mater is bound to the surface of the brain by special astrocytes projecting and sticking the pia mater in place. It goes into every fold and every crevice of the brain, and it expands or it accompanies all of the cerebral blood vessels as well to help them get into the brain and go to their special internal structures. Now you might say, well, I thought there was a blood-brain barrier. Why do we have cerebral blood vessels? Well, we still have to have the blood coming in to filter the CSF, and there are a few kind of punctures in the blood-brain barrier that we'll learn about as well. So there you see our 3 meningeal layers. Same exact idea that we were looking at in our spinal cord, except this is on our bone, on our skull. So the dural folds are going into the our deep fissures of our brain. So these are extensions of that dura mater into the cranial cavity. They go in with some of these fissures, like the falx cerebri, the tenatorium cerebelli, and the falx cerebelli. Those are the dural folds. The dural sinuses are large collecting veins within those dural folds, just below or deep to that dura mater. The superior sagittal sinus is the largest. So this is where the blood circulation, the venous return, is going to collect before it goes down back to the heart. The dural folds, the falx cerebri, this is between the 2 cerebral hemispheres. So again, they fold in and contains the superior and inferior sagittal sinuses, again, assisting with blood pooling before it goes back to the heart. The tenatorium cerebelli is separating the cerebral hemispheres from the cerebellum, and the falx cerebelli is separating the 2 cerebellar hemispheres from each other. There you see these invaginations or foldings of the connective tissue following the surface of the brain, and the sinuses, which is where the blood will pool. Sinuses aren't really a big deal yet when we get into A&P2 and we are talking about the cerebral circulation. That will be a big deal then. So let's talk about CSF. Our cerebrospinal fluid completely surrounds and bathes all of the surfaces of our central nervous system. The volume at any given time is about 150 milliliters, so not a whole lot, because it's a very small space that's occupying. The entire volume is replaced about every 8 hours. Just like everything, it's replaced and refreshed just to keep it balanced and within homeostasis. It's produced by a special area called the choroid plexus. The choroid plexus is located within the ventricles, and this is a break in the blood-brain barrier, so the blood comes in and is filtered at the choroid plexus. It's very specialized ependymal cells and capillaries there. So it circulates from the choroid plexus through all of the ventricles. From the 4th ventricle, it goes into the subarachnoid space, and then down the central canal of the spinal cord, and then it can diffuse across the ependymal cells. And that allows for the exchange of materials from the central nervous system to the CSF, so back and forth. The CSF is then reabsorbed at various locations throughout throughout the kind of root. They're called arachnoid granulations. They're little finger-like extensions of the arachnoid membrane, and they extend into the superior sagittal sinus. That's the main location of the arachnoid granulations. Here you see our choroid plexus within the lateral and third ventricles secreting the CSF. If we look at a closer view, you see that it is special capillaries within the choroid plexus that allow for the exchange of materials through special epidermal cells, leading for that exchange of nutrients and waste products. And the CSF can then circulate down through the lateral ventricles, interventricular foramen, into the third ventricle, into the cerebral aqueduct, into our fourth ventricle, into the central canal, and then through the subarachnoid space, where it can then be reabsorbed by the arachnoid granulations, which are scattered throughout, especially in the superior sagittal sinus, as you see up here, this little area. And that's where that CSF will be kind of filtered back into the blood. You can pause the video here, go back and answer these 3 questions. Let's talk about the medulla oblongata and the pons. So we've talked about some of the basics of the brain, the coverings, the ventricles, the CSF, the protection. Now we're going to start with the basics of the brain, and then we're going to get more and more complex. So the medulla oblongata, this is as basic as the brain gets. Without a medulla oblongata, you're not living. All communication between the brain and spinal cord are relayed through the medulla oblongata. They come through the medulla oblongata. There's ascending sensory information and descending motor information. Those tracks are on the anterior surface of the medulla oblongata, and it's also coordinating our autonomic reflexes and visceral functions like our digestive functions, as well as our heart rate, respiration, blood pressure, all the basics. So there are some anatomical features here. There's something called an olive. It looks like an olive. It's a bulge on the anterolateral surface, and it contours something called the olivary nucleus. There's also pyramids. This is on the anterior surface of our medulla oblongata. These are just tracts of motor fibers that are originating from the cerebral cortex. So coming down from the cerebral cortex and kind of concentrating here in this area we call the pyramids. Some of these fibers go onto the opposite side there. Discussation is the crossing over, and this is what happens either in the medulla oblongata or in the spinal cord, no matter what. So let's talk about some of the components of our medulla oblongata. You have the autonomic centers and the relay stations. The autonomic centers are controlling your vital functions. The relay centers are basically located along those sensory motory pathways, making sure the information gets to where it needs to go. There's also nuclei that are associated with all of the cranial nerves that originate on the medulla oblongata, and there's 5 of them. So here you see what I mean. Looking at the pyramids on top, they kind of look like little bubbles, not really pyramid shaped. You can see the olivary nuclei. You can see the relay centers here going up and down throughout the medulla oblongata. When you look at a posterolateral view, you can see some of these a little better, some of the different nuclei, the nucleus cuneatus, the nucleus gracilis. These again are just nuclei that are associated with specific areas of the body. We'll look at these as we kind of move through here. The nucleus gracilis and nucleus cuneatus are just relaying sensory information. The gracilis is relaying information that's more medial to the body. The cuneatus is more lateral to the body. Olivary nucleus is relaying information to and from the cerebral cortex and the cerebellum. There's also something called the solitary nucleus, integrating visceral sensory information to all of the autonomic processing centers, like so that you know where your digestion is occurring or where your food is along the digestive pathway. And then all the autonomic reflex centers and other centers like sensory motor nuclei of those cranial nerves. Cranial nerves 8, 9, 10, 11, and 12 are all relayed through the medulla oblongata. So let's talk about the pons. The pons links the cerebellum with the midbrain, the diencephalon, the cerebrum, medulla oblongata, and spinal cord. So it's basically the next stop up into the brain. We have ascending and descending tracks here also, motor and sensory pathways. This is where our respiratory centers are. This is also where our reticular formation is based. The reticular formation is kind of this kind of hodgepodge mess of gray matter. It's embedded in a nuclei, and these reticular formation is basically regulating vital autonomic functions. It's going from the medulla oblongata into the midbrain, concentrated in the pons. Here you see what I mean. Here's the pons. Looking at it closer, there's all these transverse fibers. There's ascending and descending tracts going through it. The pneumotaxic and aponeuristic centers, which we'll learn about next semester, are all in there. And here's that fourth ventricle. And the reticular formation is this little tube that goes through all of these brainstem structures. So there you see the nuclei that are associated with the cranial nerves, the aponeuristic and pneumotaxic centers, which are just relaying and regulating respiration rate and depth of respiration, relay centers for sensory motor pathways, ascending, descending tracts, and transverse fibers interconnecting all of the cerebral hemispheres, or cerebellar hemispheres, I should say. Here's the review. You can go back, pause the video, and answer these 3 questions. Let's keep going. The cerebellum is our automatic processing center. This is monitoring our body's position through proprioceptors, visual, tactile information, balance, auditory sensations, basically anything with body positioning or where you're looking, all relayed through the cerebellum. So damage to the cerebellum could be caused by trauma or stroke. Temporary damage could be through drugs or alcohol. That's why you lose your balance and get a little loopy when you're under the influence of drugs and alcohol, because your cerebellum is affected. It could result in ataxia, which is the lack of muscular coordination. In severe ataxia, the individual can't sit or stand without assistance. And unfortunately, many of us probably know someone that has been in that situation before. I was out to dinner the other night, and this guy could not even stand up. I didn't even want to be at the restaurant looking at someone like that. It was so uncomfortable. Control yourself. Anyways, let's talk about the functions of the cerebellum. There's 2 main functions. One is adjusting the postural muscles, and the other one is to program the fine-tuning movements of our conscious as well as our subconscious activities. So by adjusting our postural muscles, we're basically adjusting and monitoring how we're sitting or standing, right? This is when you're standing and you keep shifting your weight, right? Or fine-tuned movements would be like a learned motor pathway, like walking upstairs. You have to you have to bend your leg and step and transfer your weight and lift your foot, and that whole process is all done and controlled by the cerebellum. The overall anatomy of the cerebellum, there's anterior and posterior lobes, there's a primary fissure, and 2 hemispheres. The 2 hemispheres are separated by something called a vermis, a worm, and the surface is covered by gray matter called the cerebellar cortex. The folds are called the folia, and they're much less prominent than the cerebral folds. Here you see them. The cerebellum anatomy, it has an inner cortex that's inner and granular, and an outer molecular layer. There's a layer of Purkinje cells there, which are highly branched. They're getting input from many synapses, many dendrites. And again, this is because it's your body's positioning. My elbows are bent, they're here on the table, my hands are going like this, I'm sitting here, my legs are crossed, right? All of those things are all being relayed and processed in the cerebellum. It's getting all those motor commands out. Only the axons leaving the cerebellar cortex are from those special Purkinje fibers that are getting all that information. So in other words, the cerebrum is only getting kind of a summary of everything, right? It's not getting every little detail. It's kind of getting the cliff notes. I'm going to stop the video here because I hear my children calling me, and I will return with the second video in just a few minutes. Let me stop recording.